Thank you very much for your introduction. Good morning. First of all, I would like to thank Scientific Committee for giving me this opportunity to present our results from Notre Dame. And I expect that not many of you know where Notre Dame is, so I presented this map here. So Notre Dame, I would say in comparison to Toledo, which is like a mixture of three cultures, Notre Dame is on the border of three states. So it's Indiana, Michigan, and Illinois. So it's about 2 hours by driving from Chicago. And here on this picture, we have our campus. It's a very beautiful campus. I would say so, almost so beautiful like Toledo. Do you agree with me? I hope yes. However, it's much colder there. We have at least 6 months of winter. And we have also very big stadium, because maybe if you know, Americans are crazy about football, American football. So this stadium can have like 80,000 fans who comes for games. And I work at in the Department of Physics and also in Radiation Laboratory, which is in the International Center for Study of Chemical Reactions initiated by ionizing radiation. And our research is sponsored by US Department of Energy, and we deal there with basic science to understand radiation physics and chemistry. We work on solar energy conversion, nuclear energy applications, and also biochemical applications. And this laboratory, has a lot of facilities, and we have many users from around the world. We have 3 gamma sources with different activity, and we have also 3 electron accelerators, and this is one of them, very famous, again, football, because we are also crazy about American football, Van de Graaff electron accelerator. And as we could hear already on 2 previous talks, law or electron collision drives many chemical and physical processes, and they are important in many applications. Here I listed some of them, like important for our laboratory, radiation waste management, or radiation processing, radiobiology, radiotherapy, astrobiology, plasma. We could hear a lot about plasma during this session, lithography, also solar cells, explosive detection, and also we need to remember that when we probe our samples with high energy radiation, or when we use detectors when we heard from Sasha talks for detection of particles, high energy particles, we need to remember that such ionizing radiation produces a lot of secondary electrons, which can also be involved in many reactions. And my talk is about dissociation induced by electrons, or fragmentation. And on this scheme, we have different possibilities how molecules can be dissociated. So for example, we can have dissociation due to impact ionization, or due to excitation, or electron attachment. And on this talk, I will focus on one of the process, which is dissociative electron attachment process. We also, during this conference, had a couple of talks already about this process. So just shortly, I remind how it looks like. So as we have written in this reaction, electron interact, let's say, with AX molecule. It forms transit negative ion, which can have lifetime even up to microseconds. And then it decays into negative ion and some neutral fragment or fragments. And more about this process, but I will go very briefly because this slide was already shown a couple of times. So it shows the potential energy curves. So black curves represents a ground state for a molecule. And when we have electrons which interact with such an AX molecule, we can have transition to one of the excited state, which is shown by a green curve or red curve. And if we have excitation of a molecule of, in this case, already an ion, we can have auto-detachment of electron, or molecule can dissociate into fragments. And what we detect in our experiments, we detect negative ions. We detect ion yield as a function of electron energy, and we observe resonant structure on our spectrum. There are also a couple of types of resonances. These are two most often observed types, like valence resonance and dipole resonances. So in the case of valence resonance, such a low energy electron can be attached to molecule, and if energy state of such an ion lies above ground state, we call shape or single particle resonance, or if it lies below ground state, we call nuclear excited Feshbach resonance. Also, this incoming electron can electronically excite molecule, and in this case, we deal with core excited resonances, which usually occur at much higher energies on our spectrum. The second type of resonances, it's a dipole resonance, and it's formed when a molecule has a critical dipole moment around 1.6 dBi. It was predicted from theory, I think, of Fermi and Teller. However, in experiment, we observe that usually electron can form such a dipole bound state when a molecule has dipole moment of above 2 and 2.5 dBi. And here there are some examples of cross-section for DEA process. So what is unusual with this process that occurs at very low energy, here it's example for formation for different processes which involves low energy electrons with interaction with oxygen. And we can, in this case, maybe cross-section is much lower than, for example, ionization of oxygen molecule, but we have plenty of different molecules which has really huge cross-section, and this process can be very important to know and understand. So on my second, or maybe at the beginning, I showed different applications where low energy electrons are applied, electrons with energies, let's say, below 100 eV. But on this slide, I show application of the specific process, DEA process. So again, we had a couple of talks during this meeting, and we could hear, for example, that electrons are very important in radiation damage to DNA. This was talk given by Elahe Alizadeh. We could also hear that such electrons can be important in radiotherapy, which Linda Feketeova presented. During this session, we saw applications of this process in atmospheric physics or planetary atmosphere, or in semiconductor plasmas, or in general in plasmas. Also, there are many applications that people try to understand how we can synthesize bigger molecules from small molecules, and how the life was originated. And we heard talk that DEA process can be used in nanofabrication, given by Professor Ingolfsson. So typical experimental setup for this type of studies is presented here. It's a just general view of such an experiment. So of course, the main part, it has to be some electron source. It can be filament or some commercial electron gun or monochromator, from which we can produce low energy electrons. Of course, we need also target, and we study different targets in different phase. It can be gas phase or vapor, or if it's a solid state, molecules in solid states, we can evaporate molecules in the oven. And what we measure negative ions which are formed by using also different types of mass spectrometers. And this is this experimental setup in our lab at Notre Dame. It's relatively simple because it's new. We still extend functionality of such a systems. So we have just electron gun, quadrupole, of course, high vacuum chamber. We have also another electron source in front of a quadrupole mass spectrometer. So in this experiment was built by Michelle F. Dowley, former postdoc in our group, and also with big help of students from Innsbruck, Benjamin Pushnik, who helped set up this equipment. And here we have a resolution of electron beam around 0.3, 0.5 eV. However, now we are constructing new equipment where we want to improve resolution of electron beam even to a couple of milli eV. And former, again, postdoc Denis Sokolov, he started building this machine, and we are continuing development of this. So my talk is about heterocyclic molecules. So I will show you some examples of molecules which have ring structures and how the DEA process is involved, what we learn from them. So here, example for a 6-member ring, pyrimidines like uracil, thymine, and cytosine. And first, what we measured total ion yield as a function of electron energy from thymine. We could see huge signal at low energy electrons below 3 eV, and some smaller signal around 6, 9 eV. So here we measure total ion yield. But we wanted to know exactly what kind of species are formed at which energies, what kind of chemical bonds are broken in such a process. And what we observed at these low energy electrons, the most abundant ions which are formed are dehydrogenated molecules when such molecules lose hydrogen. And what we could see that uracil and thymine give very similar signature in our spectra, and the difference between both molecules is that we have additional methyl group in a thymine molecule, and in uracil we have hydrogen only. So what we learn from this that methyl doesn't influence properties and the signature of ion yield in this case. However, when we look at such molecules, we don't know exactly from which position hydrogen is removed. So we ask our colleagues from theory to calculate thermodynamic thresholds for dehydrogenation, and they calculated for us this bond dissociation and electron affinity for these radicals, and we could obtain thresholds for formation of dehydrogenated molecule. And from the calculations below 3 eV, all channels are possible. So we were still not happy because we wanted to have more information. So we came back again to experiment, and we did experiment with deuterated and methylated molecules, where we put methyl or deuterium at different positions. And doing such an experiment and by comparing shapes of ion yields, we learn that this channel only occurs when hydrogen is removed from nitrogen side. What else is that at low energy, resonances at low energies are formed when hydrogen is removed from N1 position, and this broad resonance at around 2 eV is formed when hydrogen is removed from nitrogen 3 position. So it's a very nice site selectivity in this DEA process. We try to understand formation of such resonances, so we also get help from other group to understand what's going on. So first thing, in order to form an ions, we need to form a dipole bound state, which is represented by this red curve, and lies a few milli eV below ground state of a molecule. And we can see that this dipole bound state overlaps with sigma star potential. Therefore, we can have a transition or coupling of both states and dissociation of molecule. So by looking at these vibrational excited states, and we could identify what type of resonances we observe in our spectrum. So we could assign certain peaks to a certain transition, electronic transition in the system. We observe site selectivity in the complementary reactions where we observe hydrogen and ion. And again, we could clearly see very nice that by tuning electron energy, we can cleave specific bonds. And these reactions can be very important. For example, in this case, we form dehydrogenated radical. Let's say this is for thymine. So maybe you heard from biology that UV light can create thymine dimers, which are very dangerous for our cells and DNA. And similarly, electrons can induce such a formation of radicals and then synthesis of dimers. Or also in radiation chemistry, when we have other complementary channel, we form radicals, hydrogen radicals, which are also involved in many chemical reactions. So this selectivity, which we observe for electrons, were also observed by other groups. I will mention again in a minute about this. But before, such a selectivity were only obtained by tuning thermodynamic parameters like pressure, temperature, or using photons. And this slide is from a talk of Professor Krishna Kumar, who also looked and saw site-specific dissociative electron attachment in small carboxylic acids, alcohols, or we recently published also site selectivity in amide or peptide bond. So examples which I showed you before, they are for simple cleavage, where just single bond is broken. But we also could observe some selectivity for more complicated channels. I showed you also this total ion yield for thymine that at higher energies are at 6 or 7 eV. We saw some resonance structures, and it's mainly due to formation of NCO minus fragment. And in many organic molecules, we can see formation of this fragment, and it's mainly due to high electron affinity of such a species, which is about 3.6 eV. So here we have also ion yields for thymine, for methylated thymine at different positions. We cannot see clearly here site selectivity, but what we see that resonances correspond in all cases. But what was very interesting in this experiment, that we could see site selectivity in a metastable decay. So what does it mean that, again, we use thymine for our studies, which was methylated at different positions. We blocked certain positions. We selected channels, or we selected ions which have loss of hydrogen from molecule, and we detect NCO fragments. And this is the total yield which we observe from thymine, which was not methylated at all. And when we use methylated thymine, we could clearly see and identify from at which energies we form this NCO fragment, from which part of a molecule. Recently, we started to look at more complicated ring structure molecules, like hypoxanthine. And hypoxanthine belongs to a group of molecules which are called purine, which has, again, this pyrimidine ring structure, like thymine, uracil, and cytosine, and also has additional small imidazole ring. So we look at the most abundant fragment, again, hypoxanthine minus hydrogen, and we compare the ion yields and, in general, fragmentation to this which was observed before for adenine. So in first place, when we saw this comparison, we saw, okay, it looks very similar. We have this strong peak at 1 eV, and previous studies could identify, again, different resonances. We didn't see strong peak at around 2.2 eV. In previous studies by different groups, sometimes people attributed this peak to amino group. So we said, okay, it's fine that we don't have this peak, because if we look at the structure of adenine and hypoxanthine, the difference between both molecules is that here we have amino group and here we have a carbonyl group. So we said again, but then that it's okay, but then we look at the ion yields, and here we have around 5,000 of counts per second, and here in the case of hypoxanthine, only about 30 counts per second. So we started to think everything what we learned about site selectivity doesn't work anymore. And we were in very deep troubles. So we had to go more to dig in literature to understand what's going on in this case. And what we learned from other research for adenine or hypoxanthine in the gas phase, that in the case of hypoxanthine, we deal with many different tautomers which are possible for this molecule. And here they are listed tautomers, and we calculated the energies. And what it turned from previous studies and also from calculations that the most stable tautomer is when we have hydrogen at this position of nitrogen and 7. So it's different than in adenine. In the case of adenine, we have almost 100% of just one selected tautomer, and here it's a mixture. And what was in adenine, this signal, the strong signal which was observed, it was when hydrogen was removed from N9 position. We see here only low signal due to other tautomers which doesn't have hydrogen at this position. So we were relieved that, again, site selectivity also works for more complex molecules. Again, we calculated the thresholds, thermodynamic thresholds, also the dipole moment, and the highest dipole moment it's for this type of tautomer. And such a molecule can also form very weak dipole bond anion. Currently, we are working on, again, simple 5-member rings. This is project of one of my students, Joe Lee. We want to measure whole family of 5-member rings. So far, we finished this 3-one. And again, we learned a couple of interesting things from these new experiments that, for example, we have a dehydrogenation channel quite strong from isoxazole, much weaker for oxazole. And we can see that position of these different atoms play important role. And also the DA process depends what kind of atoms we have in the ring, because in the case of thiazole, we don't see this channel at all. So again, we did some calculations with some preliminary studies, and very new thing which we didn't observe for previous molecules, that during hydrogen loss from isoxazole from this position, and due to electron attachment, we have ring opening of molecule. And this can have very important applications for different reaction in synthesis. Similar ring opening was observed for different technique, where we had electron attachment to similar molecule, however, without H loss. So now we continue this type of studies. We methylate our molecules in order to know and understand what's going on in similar systems. And in very brief conclusion, I hope now you believe that DEA process is important in many applications: radiation damage, fabrication, plasma physics, atmospheric physics, and so on. And we could see resonant features for DEA process which show selectivity towards molecular bonds, simple cleavage bonds, towards functional groups and isomerization. And also, what is important in these studies, like to understand this ring opening, we need more experimental techniques and also theoretical studies to understand this transient and ion. So right now, we know exactly what are our products, final products, but we don't know what's going on in between when we form a transit negative anion. And at the end, I would like to thank my former postdocs, Michelle Dowley and Denis Sokolov, who were constructing our setups, and my current students, and people and other members, and people who are underlying work with DEA projects. Joe Lee, we have Alexander Miloslavljevic, who is visiting professor from Institute of Belgrade, Brandon Jones, Christian Huntington, who is a high school student, and Victor Garcia, who is not on the picture. And I would like to thank our collaborators from theory, Ian Carmichael and Ksenia Bravaya, and other collaborators in Europe, Professors Paul Scheier, Stefan Denifel, Eugen Illenberger, Philippe Ferreira da Silva, and the students, Katrin Tanzer and Benjamin Pushnik, and thank you very much for your attention.